Hello, everybody, and welcome to the deeper dive discussion number 2, which deals with immune-altered development and responses of the human defense system. We're going to be talking about profile of immune cells within individuals with Kabuki syndrome. So without further ado, I am here to introduce Dr. Sarah Potter. She's going to be speaking first, and along with Dr. Brittany Simpson is going to be on the call as well. So Dr. Potter, the floor is yours.
Great, thank you very much. So today I'm going to be talking about the immune system and how our lab really is involved in looking at immune responses and epigenetics, and we collaborate with others for different organ systems as well. But today's talk will be mostly on what we have found both in our some data from patients and as well as from mouse models. So what is the immune system? It usually protects the body against outside invaders and microbes and other things like that, but it's also important for development and clearance of rogue cells that might be involved in cancer and tumors. That also is involved in organ development, and it clears dead and dying cells away. So the immune system has a lot of other kind of lesser known roles, but to focus on the main role of protection of your body from outside invaders such as bacteria and viruses, it puts up a kind of like a fortress. So there's a lot of physical barriers such as your skin and your mucosal layers. Those are your physical barriers, as well as you have chemical barriers such as your acid in your stomach can kill things. Lysozyme from your eye, so when you cry, the tears also has some enzymes in there that can kill some bacteria and viruses, as well as some biological components such as immunoglobulins, which are important for bacteria on mucosal surfaces such as your gut. And this will bind to bacteria and then they can't enter your body. And then we have like these first responder type cells. So they are surveilling your body and seeing if there's anything that looks out of place, and then they will either eat those bad things to destroy them using some enzymes within the cells themselves, or they cause a lot of inflammation. So you might get a big red welt or something like this, fever, things of that sort, that you would then kind of be alerted, your body's alerting to other cells to come fight this bad thing that has entered your body. And today I was talking most about these kind of later stage cells that then are recruited to those areas of inflammation to kind of fight more specifically these pathogens that enter your body. So there's two types of cells that are involved in this, and this is the T cells, and they can be broken down into CD4 and CD8 cells, and I'll talk a little bit more about those in depth. And then we also have these specialized cells called B cells, and they produce these antibodies similar. Some of them will be these IgA that is kind of secreted into your gut, but then there's also some floating throughout your body, and also some that stay on the B cell themselves to help with the defense against all these external things. So a little bit about what you would expect to see has been compiled by some other groups as far as immunological symptoms go within individuals with Kabuki syndrome. This is a compilation of 177 individuals that had been surveyed, but also it includes both type 1 and type 2. A large percentage of individuals have infection susceptibility. This includes most frequently the ears, nose, and throat infections, so sinus infections and things of that sort. A large percentage also have combined variable immune deficiency, and also hypogammaglobulinemia. This is basically you have a decrease in your antibodies that are being produced. And then there's a portion of individuals that actually do not have any of this infection susceptibility, so no chronic infections and no hypogammaglobulinemia. And then as far as antibodies, it has been shown by this group and also by my prior mentor, Andrew Lindsley's group, that there's not adequate protective post-immunization antibodies. So when you get vaccinated or immunized against various pathogens to help boost your protection, this actually, in individuals that have Kabuki, do not seem to have this protection over time. And also, additionally, patients have autoimmune diseases, and this actually increases with age from 13% to 25%. And just as a general baseline, and the other individuals tend to have between 7 and 9%, and as far as having autoimmune diseases in adults. And so there's basically an increase of autoimmune occurrences within Kabuki individuals. And about half of those individuals that have autoimmune disease have immune thrombocytopenic purpura, and this is caused by platelets, and they are really severely decreased, and that is usually caused by either those T or B cells that I mentioned earlier attacking the platelets. And then there's other, and this tends to be chronic compared to the general population, where only 20% of individuals have this chronicity. And then many, and some individuals also have additional autoimmune manifestations. So what is the immune system doing? It is an innate immune system. These are those first responders. They hold up the barriers. You have blocking of entry of the cells that are coming in through all those mechanisms, and then sometimes they get around the barriers. And when they can get around the barriers, the cells will find them, and they're kind of like garbage men. They will target anything that comes into the body and destroy it. And sometimes they can't handle that themselves, so they call for backup. And so what the B cells do are they either take little pieces of these invaders and show them to the T cells, and they can also have a role in the chemical messenger system. So that it's basically they're telling other people or other cells about what's going on there, and together it creates a communication. So the B cells talk to the T cells, and then they are able to produce those antibodies that are really important for fighting infections. And so there's many things that the antibodies can do. They can eat, basically a fancy word for eating the bad guy or the pathogen. They can form clumps, or they can precipitate out or neutralize those pathogens, or they can poke holes in them. So we found that previously in 2016, the Lindsley et al. Has talked about B cells are reduced in individuals with KS1, and these further characterization of these B cells are that they have decreased memory, and these are special ones that can recall if they've seen that bad guy before and respond quicker. And they've also noticed that there's decreases in that special IgA that blocks the bad guys from getting in. And more recently, Hans's group has shown in mouse models, in addition to Andrew's group, they have shown immature plasma cells, which are the guys that produce the IgA, are enhanced at the immature stage, so they don't produce as much antibody, and they don't do the nice switching into this specific type of antibody. And the Pyrus patches are decreased, and that is kind of where the immune cells kind of congregate and talk together. And then so very much clinically related is IgA is decreased, and also other types of antibodies, IgM and IgG, are also decreased. T cell major functions are there's two types. One is the special innate cells, so those ones that are first responding and they're making the inflammation and such. They will chop up the bad guys and present them to the T cells, and the T cells will then either tell more people about it, and they'll recruit more cells to the site, or they'll talk to B cells to help produce the antibody, or there's a different type of T cell that will then release some factors and poke holes into this any cell that has that infection. And so when we looked across all the different types of T cells, so we wanted to look kind of like, this is what your blood report would show. So you have just general T cells, and in the gray box area is your the general population statistics, and then based off of just individuals that were part of our cohort that were recruited. The red boxes are just noting if you have cardiac surgery, as this may be relevant for T cell output, as the thymus is, which the T cells develop in, sometimes gets removed during surgery. So we wanted to make sure that that was not playing a role, which it is not, in our results. And we find that there's not really any differences between individuals with KS1 from the general population. The special CD4 cells that tell everybody things don't seem to have any major differences, and CD8 cells do not have very much differences. But when we look further into those categories of cells, we found that usually in the these cells are either the CD4 or CD8, but there's a special type of T cell that actually doesn't express these markers CD4 or CD8, and they're called double negative T cells. And they have been characterized in autoimmune diseases. And so we checked this special subset of T cells, and we found that majority of those cells are in the individuals that have KS1. We find pretty much is ubiquitously increased in our cohort, and we find that these increased cells, and it's been described that individuals that have lymphoproliferative disorders also have this increase in these cells and an enlarged spleen. And we found we could recapitulate this also in mice that have this the loss of KMT2D, which drives KS1. And then furthermore, we wanted to look at those subsets of CD4 cells. So the CD4 cells, remember, they tell everybody about this infection, and they talk to the B cells and help antibody formation. So what happens is they form in the thymus, and then they get released. And so your blood report will show either naive cells or memory cells. And if you can see here, there's a kind of a trend of individuals that are below the general guidelines for CD4s. The naive cells, and then there seems to be an enhancement of memory CD4s. As you can see here, that most of the dots are actually increased on the upper side of the general population. And when we looked further into the naive cells, the cells that have just been released from the thymus can be noted by a special marker, and this was also checked, and we found that pretty much everyone, with a few exceptions, had some decrease in these cells that are released. And to look a little bit further about what's causing this, we looked at the development of the T cells, and we found that there's two stages, or three basic stages of what happens in the thymus. So you go through double negative to double positive stage, and this is where selection type things happen. And we find that there's an increase in CD8 single positive cells, and then when they exit From here, they exit into the periphery, and we find that there's an accumulation of CD8 cells, both at this mature stage, and then there's a mature 2 stage where it basically needs a license to leave. And so this licensed expression includes various markers to allow it to get out to the periphery. And we find that with mice that have that mimic the Kabuki type 1, that these licensure genes are decreased, and the stickiness on the gene of the cells within the organ are also decreased. And so with that, I'm sorry I covered it so quickly, but with individuals with KS1 tend to have more chronic infections and autoimmunity, and this can be contributed by both B and T cells. And the B cells tend to be immature, resulting in less IgA. And T cells, we have less of a naive T cell subset with decreased output from the thymus, and they tend you tend to see more of these specialized D and T cells with an increased splenomegaly. And so our goal of future research is to understand these changes and try to find more targeted treatment of these immune deficiencies. And then this work was performed as a collaboration between 3 groups, Artem Barsky and Andrew Lindsley's labs and Hans's lab. And these are all the people that also helped with the findings. I flew through that, but I just wanted to make a comment. The reason why I wanted to present all those clinical blood updates is so when you do have some labs that come out, you could look at these from the blood analysis, and then that may make more sense to why your any individuals with KS1 might be outside some of the normal ranges. And with that, I'll open it up to Brittany.
Hi, everyone. Thanks for that awesome talk, Sarah. So I'm just here to help, answer more clinically relevant questions as well that may come up. But I did, want to put a plug in. All of this work with Sarah and the lab and everything is fueled by patient participation. So within the Epigenetic Syndromes Clinic, our home for our Kabuki syndrome patients, we are able to, upon, of course, consenting and people who are actually interested in participating, we're able to store those samples and do work such as what Sarah showed today, and then also able to say, hey, we have all these samples, please give us money to study this, because as you have heard all day, funding is really, really difficult to acquire. And so with that, if we can show we have interest, if we have participation, it's really great. And then 2, we can see anybody who is interested in their family in the Epigenetic Syndromes Clinic, in order to, kind of provide a full scope of what is going on with you, with your healthcare, your child's healthcare, as well as, if we need to do those some of those studies that Sarah was talking about for the immune system. We do have an immune collaborator and things like that. We're trying to grow other specialties as well. But and so for the question part, Amanda or Tom, is someone going to ask us questions? Do we look in the chat?
Yeah, I could lead the session, myself and Amanda. So I'll direct it to you guys, and whoever would like to answer them, that would be great. So with that, are you guys ready for some questions that we have here? Okay. All right, so the first one that I have is the question is, if a child with Kabuki showed titers to the infancy vaccines at age 3, should that indicate that they will continue to have the protective antibodies as they mature, or should this continue to be monitored?
So that's a good question. So it really depends on what vaccine that you're referring to. We recommend all individuals have the national guideline as far as the vaccines that they get when they get them, because in everyone, your immunity falls over time. So that's why they recommend the tetanus titer every so often, the tetanus vaccine and others every so often and around childhood. We do see sometimes that individuals with Kabuki syndrome may not respond as well. And so you may need a different booster than what is generally recommended. But in general, if you do see, now tetanus is when the body's really easily able to make antibodies against. So that's kind of the low bar. So that one, it shows promise that an individual is able to make that robust response, but it does depend on the different vaccines. But importantly, we recommend them. They're very, very important to protect us from those diseases we've conquered.
Thank you so much, Dr. Simpson. The next one I have here is, what is chronic ITP and can it ever be fully resolved?
That's the good question. So that is when your platelets are being digested too quickly. So our body makes all these wonderful cells that float around in our blood in the bone marrow. And so that would be, the red blood cells, which help us with oxygen, the white blood cells, as Sarah has really outlined, as our kind of a defense fortress against these invaders, and then platelets. Platelets kind of go around and like, oh goodness, that vessel is a little leaky. We need to plug that hole. Or when you get cut, they come in and they stop you from bleeding, which obviously is really important. So with ITP, what's happened is the body just makes a mistake and is like, oh, I need to get rid of that, but it's actually your platelets. So it really varies from child to child or adult to adult as far as how their body's responding. And sometimes it can improve over time, but so it just really varies.
All right. The next one I have is around vaccines. So with the findings about vaccine efficacy or antibody formation, has there been any thoughts or theories around alternative vaccine schedules for children with Kabuki in terms of timing and age, etc.?
Very good question. To my knowledge, that has not been something that anyone has really looked at to change the vaccine schedule for individuals, but that will definitely be something we talk about with the clinical guidelines. In my experience with other conditions similar to Kabuki syndrome, like Rubinstein-Tabby syndrome, we haven't recommended differences in the vaccine timing, but just because it is so individualized. But then with this particular, with the Kabuki syndrome, we will kind of see what the international experts say.
All right, I have one or two more here. This may go back to what we had just previously talked about, but essentially it's, what do we need to tell? What was that?
I'm not sure.
Okay. The question is back to like, what do we need to tell our child's doctor? Is there a quick summary which may help them in their decision of making during primary care?
So one of the great things that you guys will be able to have in hand is our management guidelines once those are able to be published. So it is a long process. As you guys saw in my earlier talk, we've got a lot of papers to go through just in general, but also with the clinical guidelines. They're going to use some of the scoping review to actually fuel those as well, which is going to be great. The guidelines themselves are really still in the infancy phases. And so we are working really hard to get those finalized, but so it will be a year or so before those come out. That will be the best thing because that will give a very good summary for what the condition is, what the condition impacts, and how we as experts recommend you treat it. In general, actually, there is a very, very awesome website. It's called Gene Reviews. It's a public service kind of document for rare conditions. And actually, Dr. Margaret Adams, who I'm sure all of you are aware, she's a phenomenal physician out in Seattle and an expert in Kabuki syndrome. So she does gene reviews, and she actually wrote the gene reviews for Kabuki syndrome. And so that is a really good place that you can give your primary care provider the link, and because it's free for everyone. And so that goes through what Kabuki is, what do we expect as far as things that can pop up, what things should a primary care have in the back of their head saying, you know, I need to have an increased suspicion of this given this child has Kabuki syndrome or this adult has Kabuki syndrome, and then really truly how we manage them over the lifespan. So it's kind of this baby step to the guidelines we're going to be developing, but it's a really great resource. So again, that is Gene Reviews.
Perfect. Thank you so much. Let's see. This is a question. Is my daughter sees an immunologist? Are there any immunology labs that should be treated or evaluated differently due to Kabuki syndrome? Or are there standard values also valid for patients with Kabuki?
Great question. So if your daughter's seeing an immunologist, she is seeing an expert in the immune system, and the labs that they order are going to be similar to what we order with individuals with Kabuki syndrome. And so you know, they may not, if they don't have a lot of experience with Kabuki syndrome, they may not understand some of the more nuanced things to look to think about as far as like why it's happening. But I don't think that there is any difference between you know, the things that they would need to do if they're able to do that primary screening, like seeing antibodies and those kinds of things. I am not an immunologist, just as a disclaimer. So they have a lot more expertise than I do when it comes to kind of checking those labs. But we use the same labs to kind of screen to see what may be going on with an individual.
I would like to just share that we Brittany also is on this paper. We've put it on a free source called bioRxiv, and so obviously I would refer to your immunologist to use those labs that you have. But all this data that I just showed today are publicly available on that paper on bioRxiv, and we can provide that link. It's in my PowerPoint slide, but we can have maybe KSF to also share to whoever's wanting to know. But you can have them refer to that to just as a general thing, that if they're concerned about maybe a lower one of the specific T cells compared to the guidelines, you can kind of maybe refer to the fact that that would be something that is kind of known within the general population of Kabuki, that you might have a lowered naive T cell, or a recent thymic immigrant, or if you aren't responding to titers, there are those prior publications that can also be discussed with the primary or the immunologist.
Thank you guys so much. So with that, we are out of time. So I'd like to thank Dr. Simpson and Dr. Potter for this breakout session, and we are going to now return to the general session. And thank you guys for your questions. They were fantastic, and look forward to hearing some more. So thanks, everybody.
Thanks, everyone.